case anything happens. Okay, everyone guys, take a seat. We're gonna start. Come on in. Welcome. Oh. Hello. Test 1, 2. Hello. Oh, there it is working. Okay. Someone wants to join. Test 1, 2. Admit. Okay, there we go. Guys, there's plenty of room over here, so squeeze in that way to the right, or your left, your right. Sorry. Is it bigger? Yeah. Guys, come on, this guys. There's plenty of space over here. Plenty of space. Don't be shy. Say hello to your neighbor. Do you guys want to? No? Anyone else? Okay. Well, first off, thank you guys all for coming. How many of you know why you're here? Okay, good. How many of you know what decentralized science is at all? Yes? Some people? There's some people? Okay, yeah, this is decentralized, this is centralized science in this vicinity here. How many of you have been to a DeSciNYC event before? Oh wow, so many people. We have a lot of return DeSciers. We're gonna issue a token at some point, which will be cool, like a DeSciNYC token, so you can memorialize your being on the blockchain. Cool. Well, without further, let's just do a quick picture. Just, we'll do a panorama one. Oh, okay. Smile. This will go on the Twitter and such. Okay. Oh my gosh, I think this might be one of our largest ones. And let me just do one more. Okay, so we're going to do it slightly differently this time. So Michael Levin is going to speak first. How many of you have seen Michael Levin on on one of his talks already? When we see, wow, that's a lot of people. Okay, one more. When I was promoting this event, we got a lot of DMs who said he's my favorite speaker. Can you hear us, Michael?
Yeah, I can.
Okay, good. He said this is one of his favorite. A couple people messaged me saying Michael Levin is one of our favorite tech speakers that they've ever seen. So that was just totally cool. Can you see the audience here, Michael?
Yeah, I can.
80 people. It's great.
Yeah.
And then what else did I want to say? There's lots of things. Oh yeah, if you guys post us on Twitter or on Instagram, we'd be forever grateful. It helps to spread the word. It helps to decentralize science. It helps to get the word out. So I'm going to let Michael speak. I'm just going to say one thing. If you like decentralized science, we started a decentralized science company. I'll talk more about it in a second, but I know some people are going to go. But I just want to say, if you're looking for a job, we're hiring to do a decentralized science platform. So if this is interesting to you, we have an in-person office in New York City. We're looking to hire community managers, developers, business people. We pay really, really well, and we have a great office and a great team, and we're looking to grow. So if you're interested in this, and you're interested in decentralized science, maybe you're an angry academic, maybe you're looking to change the world, maybe you're looking to see something better, we'll do this. But I just wanted to get that before, and I'll do a little talk on what is decentralized science after Professor Levin speaks. So without further ado, Professor Levin is a professor at Tufts University, super renowned in his field for his work on bioelectricity, which was when I first heard about it, I was amazed, and I'll let you take it from here.
Yeah, thanks so much.
Let's give him a round of applause.
For the invitation and that very kind introduction, and thanks for letting me share some ideas with you. So let's see, I'm going to figure out how to share a screen here. All right, so I'm going to turn on my slides, and when I do, I will not be able to see you, so if anything goes wrong, just please somebody speak up. Otherwise, all right, here we go. Okay, so what I'd like to talk about today is the field of diverse intelligence. and specifically the kind of cognitive processes that go on in your body and what this means for various other issues. You can find all the primary work, like the primary papers, data sets, software, everything at this site, and you can sign up for announcements and get some thoughts sort of beyond what I can say in peer-reviewed publications. You could get that in my blog here. So there's four things I'd like to talk about today. First of all, I'm going to talk a little bit about what I mean by diverse or unconventional intelligence. I will give you some examples of such, looking at bioelectricity and how it functions as a kind of cognitive glue that binds the cells in your body into a kind of collective intelligence. And this collective intelligence solves problems in anatomical space, also known as morphospace. We'll talk about some biomedical applications that result from thinking in these kinds of ways. And towards the end, we're going to touch on some issues about the future and what I think it means for all of us. So this is a well-known classic painting. It's called Adam is Naming the Animals in the Garden of Eden. And I find this very interesting because I think there's one profoundly wrong aspect of this old story, and also there's one very deep aspect that I think is really right on the money that we have to absorb. The part that I think is wrong here is that this gives you the idea that these animals are discrete natural kinds, right? You look at this and you say, okay, I know what all these animals are. There's a finite discrete set of individual animals with specific shapes and specific behaviors, and Adam is actually different than all of them. He's special, and so I think those two things we're going to have to blow up in the next couple of decades. That will I think that's going to disappear. The part that I think that's really deep here is that in this old biblical story, it was actually Adam's job to name the animals. The angels couldn't do it. God couldn't do it. Adam was the one who was supposed to name the animals. And I think that's for two reasons. One is that he's the one that's going to have to live with them. And the other is that in these old kind of mystical traditions, naming something or knowing the name of the real name of something means that you've understood its true nature. And that understanding is supposed to give you power over that thing, right? So knowing the name of something is really deep. And I think this is important because we are all going to have to name in this kind of meaning. We're going to have to name a whole large set of novel beings that have never been here before, with whom we are going to share our world. And I think that's really important. So you no doubt have read all kinds of works of philosophy and other more modern maybe stories about the difference between humans and maybe machines and other things. And what people typically have in mind when they say those things is this kind of this typical adult modern human that has this like agential special glow about them. But we've known now for a really long time that both on an evolutionary timescale and also a developmental timescale, there is actually a continuum of bodies, and we'll get back to this towards the end of the talk. But actually, this form has very slowly and gradually evolved from all these other forms and originally a unicellular microbial ancestor. And the same thing is true during your lifetime. So whatever properties this kind of being has, and so memory, true goals, preferences, understanding, moral worth, responsibility, and so on, we really, as scientists and philosophers, we really owe a story about how that arises. Where in this continuum does that actually show up? And is it better to think about that as a kind of discrete thing, you have it or you don't have it? Or what I think is that it's actually a continuum. The right question is what kind and how much? And so I've been working on a framework to enable us to recognize, create, and ethically relate to a really diverse set of intelligences that are very different from the kinds of things that we usually recognize as intelligences. So apes, birds, maybe an octopus or a whale or something, but all kinds of other creatures, colonial organisms, and swarms like beehives and ant colonies, synthetic biology, engineered new life forms, AIs, whether robotic or purely software, and maybe at some point aliens. At some point, maybe we find some exobiological life. So we need to be able to talk about all of these things simultaneously. What do they all have in common? And I'm not the first person to try for a framework like this. This is 1943. Wiener, Rosenbluth, and Bigelow tried for a scale that goes all the way from passive matter, so basically simple passive materials, all the way up through these kinds of different stages to the kind of
complex second-order metacognition that humans have, where they can think about themselves and they know that they know things and so on. So what we need is a story of scaling. How do very simple information processing kinds of acts become minds? And whatever framework that we come up with, it has to move experimental work forward. It can't just be philosophy. It needs to actually give us new capabilities, for example, new biomedicine. And it also has to enhance ethics. It has to enhance our ability to understand our world and relate to it with the appropriate kind of compassion. So the fundamental fact is that all of us take this journey. We've all made this journey from what people call just physics, and I really hate that term, but people use it. As an unfertilized oocyte, you were once, all of us were once a little blob of chemicals. And when people look at that blob, they say, okay, well, this is just a physical chemical system. It obeys the laws of chemistry. There is no mind here, just a bunch of chemicals. But at some point, slowly and gradually, it turns into one of these amazing kinds of creatures, or even something like this. Here's Rene Descartes, who was fond of saying that he was not a machine and had special properties. And so that's fine, but we need to really then tell a story of where does that happen. And developmental biology offers absolutely no place where you can draw a bright line and say, okay, before that it was just a bunch of chemicals doing the things that chemicals do, but now, bang, now it's a real being with a real mind and preferences and goals and so on. There is no spot like that in developmental biology. So we really need to understand the scaling this. Well, at least, okay, so that's a little. That should be, if you take that on to really understand that, it should be a little disturbing. But at least we're a true unified intelligence, right? We're not like an ant colony, which people do describe ants and termites as a kind of collective intelligence, but we're not like that, right? Like we are a centralized, unified mind. We have a brain and everything. Well, Rene Descartes thought so, and in particular, he was fond of the pineal gland because there's only one of them in the brain, and he felt that our unified human intelligence should have one spot in the brain that isn't duplicated the way that other structures are. But if he had access to microscopes, he would have discovered that actually there isn't one of anything, because in the pineal gland, this is what it looks like. There are huge numbers of individual cells, and if he had really good microscopy inside each one of those cells, he would see this. This is what's actually inside. All of this stuff is inside every cell. So actually, all of us are collective intelligences. Even we, no less than ant colonies and beehives and flocks of birds. We are all made of parts. And in fact, I think all intelligence is collective intelligence. In fact, for us, this is the sort of thing we're made of. Now, this happens to be a free-living organism known as a lacrimaria, but it's a single cell. And you can see the amazing competency it has at its local scale. So with respect to single cell events, it handles all of its anatomical needs, its metabolic needs, its physiological needs. No brain, no nervous system, no stem cells. Everything is handled by this amazing little organism. And we are a large collective of cells that have enormous competencies in different spaces. And so if you look at our structure, not only are we a set of nested dolls consisting of various chemicals and subcellular components, and then tissues and organs and organisms, and then of course societies and swarms, not only structurally are we a collection of these hierarchical levels, but actually each of these has its own competency, its own intelligence, its own problem-solving capacity. So in other kinds of spaces, not just in three-dimensional space that we're used to, such as, for example, when mice run mazes and so on, but in other spaces, the space of physiological states, the space of all possible types of gene expression, the space of all possible anatomical configurations. In all of these spaces, the different levels of your body structure are solving problems, pursuing goals, storing and utilizing memories, and navigating those spaces with great ingenuity. That amazing architecture, this multi-scale competency architecture, which currently our technology, robotics and our typical AIs don't use anything like that. No reason why we couldn't, but no one's done it yet. But biology uses this amazing multi-scale architecture, and it allows some truly astounding capabilities for living forms. I'm going to show you a couple of interesting examples. One of the things that biology can do is have a kind of, you can almost call it a kind of beginner's mind concept, where it doesn't really evolution doesn't just make organisms that are solutions to specific problems. It actually makes problem-solving machines that come into the world with massive plasticity, ready to take on novel configurations. So what you're looking at here is a tadpole. Here's the mouth, here are the nostrils, here's the brain, here's the tail, this thing is the gut. And what you'll notice here is that what's happening here is that there are no eyes in the primary location where the eyes go, but there is an eye that we induced on the tail. And what you see is that these animals, so we built this machine to train these tadpoles in a visual task, and what you find out is that they can see perfectly well. And this eye, which is now located on their tails, doesn't connect to the brain. Here's the optic nerve. Sometimes it connects to the spinal cord like this, sometimes to nothing at all, and they do very well in visual assays. So just to think about what this means. For millions of years, this brain was perfectly adapted to take visual input right here from the eyes next to it. And now you've got this weird itchy patch of tissue on your back that's providing some sort of electrical signal onto your spinal cord. Why doesn't it take many rounds of evolutionary adaptation, mutation, selection for this to work? It works right out of the box. First time you do it, it doesn't require any more evolutionary cycles, and that's because this kind of creature, as all of us, has massive plasticity. There are lots of other interesting features. For example, these are planaria. These are flatworms, and what's interesting about these guys is that they are incredibly regenerative. So you can cut them into many pieces. Each piece will regenerate and become a perfect tiny little worm. Now, one thing that James McConnell discovered in the 60s, and he got a lot of flack for it, but he was actually right. We repeated his work using modern automated methods in 2013, and he was completely right that if you train a planarian, for example, to feed on these little bumpy circles, and then you amputate their head, including their brain, and you get this tail left here. The tail sits there, doesn't do much of anything for the next 8 days, doesn't have any behavior. But what it does do is it grows back a new brain. And as it grows back a new brain, you then can test these animals, and you find out that they've remembered the original information. So not only is the anatomy regenerated, but so are the memories. And actually, presumably they're stored in a tail somewhere, we don't know where, and kind of distributed throughout the body. And then they are imprinted onto the new brain as the brain develops so that they can demonstrate behavior. So the movement of information through the body, right? You're seeing here simultaneously the ability to restore anatomical structure, so anatomical memories, but also behavioral memories, and these things are linked. And one of the most remarkable examples is this. These are caterpillars, and they have a particular brain suitable for crawling around using the soft body architecture in a two-dimensional world. And they need to turn into this. This is a flying, hard body kind of being that has a completely different life, and it has a different brain because of that. In order to do this, it basically destroys most of the brain during metamorphosis. So many of the cells are killed, the connections are broken, and it refactors into a new brain. But what has been found is that if you train these caterpillars on a particular task, the butterflies or moths that result still remember. So there are two fascinating things here. Not only is it amazing to ask where is the information that it's robust to this incredible refactoring of the memory medium. We don't have any technology like that. If you start shuffling around the pieces of your memory chips inside your computer, nothing's going to work. But not only is the memory robust, you actually, if you think about it, you realize that you actually can't just store the information. You have to completely reinterpret it for a new context. For example, if you teach these caterpillars to eat leaves when you provide a certain color stimulus, those specific memories are totally useless to the butterfly because butterflies don't eat leaves and they don't move around the way that caterpillars do. They have to reinterpret those memories in a brand new way suitable for this creature and its three-dimensional new life. So during these kind of transformations, you see that memory, biological memory, isn't just stored, it's actually actively, creatively reinterpreted. These memory engrams that are made are reinterpreted for their new lifetime. So lots of fascinating things to be said about behavioral memories and so on, but I want to spend a few minutes talking about what we study, which is primarily anatomical memories. So let's think about this. This is a cross-section through the human body. So look at this incredible complexity of all the organs in the torso. So everything is where it needs to be in the right place, the right direction, next to the right thing. almost all of the time. Where does this pattern come from? Because we start life as a collection of blastomeres, and eventually these cells build something like this. Where is that pattern? People might be tempted to say DNA, but we can read genomes now, and we know that is not what is in the genome. What's in the genome are specifications of proteins, the tiny molecular hardware that every cell gets to have. But this structure is the outcome of the physiological software that these cells are executing. And so this information is no more in the genome than is the actual structure of the colony of ants and termites, or the specific structure of a spider web in the DNA of the spider. So we have to really understand, okay, how do the cells know what to make? How do they know when to stop? If something is damaged, how do we convince them to repair it? And actually, which I'll show you a little bit towards the end, what else is that hardware willing to do? What else can it build? What can we make from the exact same cells with the exact same DNA? And I like to think of kind of the end game of our field here as this. Someday, you're going to be able to sit down in front of a computer and use something called an anatomical compiler. What's going to happen here is that you will draw the animal, plant, or biobot, organoid, whatever, that you want at the level of anatomy, not at the level of genetic circuits or anything like that, but the actual anatomy. And what the system ought to be able to do is to compile down that anatomical description into a specific set of stimuli that have to be given to cells to build exactly what you want to build. Now, the practical reason we need something like this is because, imagine if you had the ability to tell groups of cells what to build, you would do away with birth defects. You would be able to heal and regenerate after traumatic injury. You could reprogram cancer. You could stop and reverse aging. You can stop degenerative disease. All of these things, all of these major problems of biomedicine would go away if we understood how to get a group of cells to build exactly what we want them to build. Now, the key here is that this device is not a 3D printer. It's not about micromanaging cells and trying to print body structures. It's a communications device. It's a way to translate your anatomical specification, what it is that you're looking for, into the goal states that these cells are accessing when they build some particular structure. Now, I'll tell you that we don't have anything remotely like this, and we can only do this in a few individual cases. And the reason is that here's where current state of the art is. We're very good at manipulating cells and molecules, processing information like this about which genes interact with which other genes, which proteins attach together, and so on. But we're really a long way away from what we really want, which is complete control of large-scale form and function. Somebody is missing a limb, we ought to be able to tell those cells to rebuild. So why is that? And I think it's because molecular medicine has been largely stuck where computer science was in the 1940s and 50s. This is what it looks like that programming a computer used to look like. You can see what she's doing. She's interacting with the hardware. She's rewiring the machine to get it to do something. And you would all laugh if I told you that on your laptops, nowadays, if you wanted to go from Microsoft Word to Photoshop, you would have to get out your soldering iron and start rewiring. Yeah? The whole point of what we've learned in computer science and information technology over the years is that if your hardware is good enough, and the biological hardware is definitely good enough, what you have here is a reprogrammable medium that is much better off controlled by communication, by signals, by stimuli. And this is what we're still missing to a large degree in biomedicine. We understand the hardware more or less. We really have only begun to scratch the surface of the software, the software of life, the intelligence that's inherent in every layer of our body systems. I'm going to show you a couple of examples of this. Well, first of all, we all know that development is reliable. So most of the time, the egg will give rise to a bunch of cells that build the correct human target morphology. But it's not just about emergent complexity or reliability. It's actually much more interesting than that. Because if you start cutting early embryos into pieces, this is naturally how you get twins and triplets and quadruplets. You don't get half bodies, you get perfectly normal monozygotic twins and various multiples. And this means that in this anatomical space where you go from different starting states, here's one, from different starting states, all of them can navigate that space to a particular ensemble of goal states corresponding to valid human anatomies. And sometimes they can actually avoid all kinds of local minima, meaning perturbations that you give them along the way. They can solve problems and get where they need to go. This is William James's definition of intelligence, the ability to get to the same end by different means. This is what I mean by different types of intelligence, by navigating these problem spaces. Now, some animals can do this throughout their lifespan. So here is an axolotl, and these amphibians can regenerate their eyes, their jaws, their tails, their limbs, portions of the heart and brain. And what that looks like is when they bite each other's legs off, no matter where that occurs, whatever the piece is remaining, these cells will build exactly what's needed, no more, no less, to make a perfect salamander limb, and then they stop. And that's the most amazing thing about regeneration, is that it knows when to stop. And so this idea that you have a particular structure, and when you deviate from that structure, the cells work really hard to get back to that same region of anatomical space, and then they know that they've got there, and they know that that's their goal, and then they stop. So that is a kind of anatomical homeostasis. It's the same thing that your thermostat does at home, where you set a set point, and when the temperature goes off, the system will work hard to get you back to that goal state, and then it stops. Okay. And by the way, this is not just this kind of regeneration is not just for amphibians. The human liver is highly regenerative. Even the ancient Greeks knew that. I have no idea how they knew that, but they did. Deer every year will regenerate huge amounts of bone and vasculature and innervation when they regrow their antlers, up to 1.5 centimeters of new bone per day they grow. And even human children can regenerate their fingertips. So if you just keep it clean, then these kind of amputations in kids end up with a very nice cosmetic outcome. So, but we're of course not as good as salamanders or planaria. So I want to show you just kind of one of my favorite examples to really try to bring home just how much amazing creative problem solving these systems have. This is a schematic of a cross-section through a kidney tubule of a salamander. And what you see here is that about 8 to 10 cells work together to build the structure, and then they leave a lumen in the middle through which the fluid has to go. One thing you can do is you can force these cells early on to have way more genetic material than they should. And when that happens, the nuclei get bigger and the cells get bigger to accommodate. When that happens, the resulting newt is actually exactly the same size because the cells adjust. Fewer of these larger cells are now getting together to make exactly the same lumen. The most amazing thing happens when you make these cells truly gigantic. Then it uses a completely different molecular mechanism such that only one cell wraps around itself, leaving a hole in the middle, and still gives you the exact same structure. So think of what this means. If you're a newt coming into the world, you don't know how many copies of your genetic material you're going to have. You don't know how big your cells are going to be. You don't know how many cells you're going to have. You have to figure out, you have to use all the tools at your disposal, all the molecular mechanisms that you have to solve this problem. How do I get from being an egg to being an adult, despite all these novelties? You can't count on anything. You can't assume that your evolutionary history has prepared you for knowing what to do. And the same thing happens in the transition from tadpole to frog. You know, they have to rearrange their whole face. But these Picasso tadpoles, which have the organs in abnormal locations, so the eyes on the back of the head, the mouth is off to the side, these guys still make quite normal frogs because all of these organs will move in unnatural paths to get to where they need to go to look like a good frog, and then they stop. So this is this means that genetics does not actually give you some kind of hardwired automatic process that just goes in the same direction. What it does encode is a machine that can do an error minimization scheme. It can actually get to where it needs to go despite all sorts of things going wrong. So now this raises an obvious question: how does it know what's right? How do these things know what a correct frog face looks like? So we've been studying this. And you know, the sort of uncontroversial example of collections of cells storing a goal state for behavior is what happens in the brain. So the way it works in the brain is that you have these electrical networks of cells that communicate electrically via ions that go in and out of these little ion channels, and then they pass through these electrical synapses. And the computations that are processed by that network result in the software that we see as behavior. So basically here, this is a living zebrafish brain that the commitment of neuroscience is that if we knew how to decode all of this electrophysiological activity, we could read out the thoughts, the memories, the goal states, the competencies of this animal. Well, it turns out that this is not anything special to or unique to brains. Every cell in your body has these ion channels. Most cells have electrical connections to their neighbors. And evolution discovered that these electrical networks were good for processing information a really long time ago, back around the beginning of bacterial biofilms, actually, long before multicellularity or brains or nerves or anything like that. So then one can ask, well, if the electrical networks of the brain think about moving you through three-dimensional space, what do the electrical networks of the body think about? What were these cells thinking about before there were brains? And what we've been studying is this idea that what they're thinking about is how to navigate anatomical space. They're thinking about how to get from whatever situation they happen to be in, whether a normal egg or some kind of perturbed system, into the right kind of body for the environment in which they find themselves. So in order to do this, we've developed some interesting techniques using voltage-sensitive fluorescent dyes to really literally read the mind of the body, to see all the electrical conversations that cells are having with each other. So these are not computer models; these are real. These are some cells, and this is an early frog embryo, and the colors are signals from a voltage reporter dye that enables us to see what all the electrical states are. We are reading it. We then have all these computer models that we use to try to understand and try to decode what are the memories here. And I'm going to show you some examples. This is an early frog embryo putting its face together. It's quite dynamic, but here's one frame from that video, and you can see the bioelectric pattern here. We call this the electric face because it basically looks like a face. Here's where it knows the eye needs to go. Here's where the mouth needs to go. Some other organs out here. This is where the organs are going to be in the future. This is a pre-pattern that it uses to figure out where to put all the craniofacial organs. Now, it's not just cells within an embryo that communicate, but actually individual embryos communicate with each other. So you can see what's happening here in the group, that when this guy sets off a wave, they all feel it. And here are a bunch of cells where you can see there's some neural firing here, but there are also some lots of activity in these non-neural cells. Okay, so these are pretty ubiquitous. These electrical networks are pretty ubiquitous. Now, observing these amazing bioelectrical signals is fun and everything, but you don't know that the patterns mean anything unless you can functionally alter them. You alter them and you see what happens. So we've developed tools mostly borrowed from neuroscience. And I think one of the lessons we're learning is that neuroscience is really not about neurons at all. The deep lessons of neuroscience tell us about all cells and about intelligence more broadly. And what we do is we change these bioelectrical patterns. We don't use, there are no electrodes, there are no magnets, no waves, no frequencies, no electromagnetic radiation. What we do is we hack the normal interface that cells are using to hack each other's behavior, which is the ion channels on their surface and the gap junctions that they use to exchange electrical signals. This is what we target. So we control how cells communicate with each other electrically. And when you do this, you can do some really cool things. For example, by injecting some RNA and coding a potassium channel, which changes the voltage of the cells, we can induce a little spot in this embryo that looks just like that eye spot in that electric face. And when you do that somewhere else, for example, in cells that are going to become gut, what you've just done is tell these cells, build an eye here. You have not said anything about how the eye should form, what are the components of an eye. You haven't said anything to the stem cells or to the specific genes inside of each one. You're talking to the collective intelligence of cells that are trying to navigate morphospace. And what you've said is go to the region that corresponds to making an eye. And they know how to do this. Here are all the internal tissues of these eyes, so lens, retina, optic nerve, all that. And they even have some other cool properties that you find in other collective intelligences like ants, which is that, so here's a lens sitting out in the flank of a tadpole somewhere. If you inject just a few cells, not enough to really make a whole eye, so the blue ones are the ones we injected, what they will do is recruit their neighbors. They tell their neighbors that, hey, you need to participate with us in making this eye, and they instruct them. So we instruct these cells, you should make an eye. They then instruct their neighbors. So all of these competencies, knowing how to make an eye, knowing how many cells there are, and recruiting new cells if they need to, All of this is built in. It's part of the competency of the material that we're dealing with. And we have extended this towards some regenerative applications. There are huge numbers of people living with limb and organ loss every day. We need therapeutics that are not just going to be for wound healing, but actually to regenerate whole organs. And we can see here that what happens here, normally frogs, unlike salamanders, do not regenerate their legs, but we've come up with a cocktail that triggers not only pro-regenerative gene expression here, but actually within 45 days, you've already got some toes in the toenail. But in our latest experiments, what happens is that the treatment itself, the bioelectric cocktail, is applied for about 24 hours. After that, you get a 1.5 years of leg growth in the adult frog. You don't touch it at all during that time. This is not about micromanaging. This is not about controlling the stem cells or 3D printing anything. This is about telling the cells very early on, go down the leg regeneration path, not the scarring path. And so eventually you get a quite nice leg that is touch sensitive and motile. We are in the process of moving this to mammals now. I should say that I have to do disclosure here because Dave Kaplan and I have a company whose job it is to try to move this into mammalian models and hopefully patients. And David's group makes these wearable bioreactors that apply the ion channel drug payload. And come back to the planaria for a moment, because there's something really interesting here about how we reprogram these pattern memories. And I want to again show you the plasticity of the material that we're dealing with. So here are the planaria that I was telling you about. Some amazing properties that we can talk about if people have questions, as far as cancer resistance, immortality. I mean, these guys literally don't seem to age, and so on. But one of the things that we need to understand is when if they were to be chopped into 3 pieces, so you got this middle fragment here, it 100% of the time it grows 1 head and 1 tail, and you get a normal planarian. So now the question is, how does it know? How does this thing know how many heads it's supposed to have and where the heads are supposed to be? So it turns out that there's an electrical circuit that controls this. We've characterized that circuit, and we've learned to rewrite it so that what happens here is that this bioelectrical pattern, so this is the memory, this is literally the memory of the collective intelligence of the body as far as how many heads it's supposed to have in here, 1 head. So what we can do is we can change that pattern to say, 2 heads. And when you do that in this animal, after you amputate, then it goes ahead and makes a 2-headed animal. This isn't Photoshop or AI or anything like that. These are real animals. And this bioelectric pattern is not a map of this 2-headed guy. This pattern is the pattern that we incepted into this 1-headed normal worm. It's anatomically normal. The molecular gene expression is normal. Here's where the head genes are, but it has a different internal representation of what a correct planarian should look like. We've rewritten the goal, the target state. So this is not micromanaging the cells. This is changing the mind of this collective intelligence as far as what I should do if I get injured in the future. So it's really a counterfactual memory. And one planarian body can store, a normal planarian body can store at least 1 of 2 possible representations of what a normal worm should look like. And in fact, one of the most remarkable things is that once you do that, the memory stays. 2-headed worms cut again and again in plain water will continue to regenerate as 2-headed animals. It's permanent, even though there's nothing wrong with the genetics of this animal. We have not touched the genome. There are no transgenes. This is purely bioelectrical. I showed you exactly where the electrical memory is stored and how we can see it. And here are these 2-headed worms. And not only can we change the number of heads, but we can actually change the shape of the head to that of other species. So the shape of the head of this triangle species can become flat like a P. phelina. It can become round like an S. mediterranea. The shape of the brain changes like these other species. The shape of the position of the stem cells does. So in this anatomical morphospace where you have attractors, you have these deep attractors corresponding to other species. They live there naturally, but you can get a different species, such as this guy, to visit these other attractors because it's so reprogrammable. Not only that, you can take it further and find regions of morphospace where machine learning folks would call this a latent morphospace, where you can build things that are not normal planaria at all, things that are spiky or cylindrical or combinations like this. So what I'm showing you is that the body is incredibly plastic. It's reprogrammable at the level of physiology, not necessarily the genetically the specified hardware. But we are not the only bioengineers that can hack this kind of morphogenetic navigation. This is what you normally see. Acorns make something like this, and you kind of get this is extremely reliable. This happens so often that you get the idea that, okay, well, the oak genome just knows how to do this. This is what the oak genome knows how to do. That is what they do every single time. But there's this amazing bioengineer, this wasp, and it's a parasite. And what the parasite does is using some kind of chemicals that are still poorly understood. It basically hacks the surrounding plant cells to build something completely different. Would you have any idea that these the nice flat green leaves with a normal genome, a completely normal genome, were capable of building something as crazy as this? We'd have no idea. When people talk about developmental constraints, there's a lot of constraints in our thinking about the process, but I'm not sure we understand morphogenetic constraints at all. I think they're much broader than people think. And this kind of hacking is ubiquitous in biology, and we are just learning to do it. And with this notion of what lies beyond the normal morphology, repair of the typical anatomy, but what's beyond that? I want to show you this last example, which is this. If I showed you this little organism and I would ask you to guess what this is, you might say we've found this at the bottom of a pond somewhere. It's some kind of primitive microorganism. If you were to sequence the genome of this, what you would get is 100% Homo sapiens. This is, we call these anthrobots. They are made of adult patient tracheal epithelia. So adult, often elderly patients donate their tracheal cells. We have a protocol that lets these cells reboot their multicellularity and live an entirely new life that has new shapes and behaviors. They look nothing like any stage of human development. And one of the unique things that they know how to do, which is really shocking and surprising, is that they know how to heal neural wounds. So if you put down some human neurons in a dish and you put a big scratch through them, these anthrobots will sit down in this kind of like superbot cluster. And what you'll see when you lift them up 4 days later, what they've done is they've taken the two sides of the neural wound and they knit them together. Who would have ever thought that your tracheal cells, which sit there quietly for decades in your airway, actually, if given the opportunity, make a motile little creature that runs around on its own and has lots of interesting abilities, such as, for example, healing neural wounds. And so one of the biomedical aspects of this is that this is a personalized medicine where cells from your own body, not genetically modified, there's no reason to genetically modify them, can be used to heal your own body. And no need for example, immune suppression, because they're your own cells. They're not some sort of heterologous cell type. So this kind of agential biomedical intervention, these kind of biobots made from your own cells to heal, are, I think, a huge potential application for the future. So the last thing I'll just say is that, not only do we have these two continua that remind us that we are on an unbroken, slow continuum with all kinds of other unusual forms, including cells. And whatever you say about adult humans, you have to ask, what can you say about some of these other intermediate forms? But also there's another continuum here, which consists of now rational changes that are made for various kinds of modifications and augmentations. So both technological and purely biological. I think that the future belongs to a kind of freedom of embodiment. I think that this standard form that evolution just sort of happened to drop us off in, that is susceptible to degenerative brain disease and limited IQ and limited lifespan and susceptible to all kinds of the dumb bacterial infections and lower back pain and bad vision and all these things. I don't think we have any particular commitment or allegiance to any of that. We are going to be improving some of the bad hardware and some of the bad firmware that evolution has left us with. And at every level of organization, you can think about augmenting these systems that were designed by basically a random walk with selection with rationally designed materials. So I think that in the future, all of you young people, and I think even I will get to see a bunch of this, I think, will be living with a huge number of alien bodies and minds. We already have some people that are some degree of cyborg. We will have hybrids and chimeras and synthetic life forms, and because of that kind of plasticity of life that I showed you, that interoperability is huge. So pretty much any combination of evolved material, engineered material, and software is some kind of living agent. And all of these things are going to have diverse bodies and diverse minds. And we really better step up and go well beyond very ancient and crumbling rules of thumb about how we deal with each other, in terms of what are you made of, and how did you get here? Those are the kinds of things that we ask when we try to understand, is something a quote unquote machine that we can do whatever we want with, or is it a living organism to whom we owe certain kinds of moral stances? Those categories are going to go out the window, because there are going to be all sorts of forms that challenge those superficial categories. And what I think we're looking for is a kind of ethical synthbiosis with a bunch of life forms that are nowhere on the tree of life with us. So I'm going to stop here. And just say that if anybody's interested in diving more into this stuff, here are some kind of academic papers. There's much more sort of general purpose writings on this kind of thing at my blog. I want to thank the people who did all the work. So these are all the postdocs and the grad students who did the biology that I showed you today. We have lots of amazing collaborators. None of this is possible without our technical support personnel, our funders. Again, I have to do a disclosure. These are the companies that support our work, and most of all, the model systems, the animals and the microbes and so on, because they teach us everything here. So yeah, thanks very much.
Thank you. Okay, I think we have time for some questions. So we will start here.
Thank you. This was very interesting. If I understood correctly, you state that this morphological plasticity and regenerative plasticity is not really encoded in our genome, but more in the bioelectrical properties and activity of the cells or the organism. However, the bioelectrical properties of an organism or of a cell depend on the channels, voltage dependent or independent channels that are expressed by a given cell, which are encoded in our genome. So in a way, the bioelectrical properties and therefore all the morphological plasticity and regenerative capacity is encoded in our genome.
Well, let's think about it. So certainly you need the hardware for any of it to work. So without the hardware, without the channels, none of this is going to work. But encoded is a funny word. Imagine, I don't know if you've ever seen this thing. It's called a Galton board. You can get it on Amazon for about 20 bucks. It's a toy. What it is, just imagine a board, and you bang a bunch of nails into it, right? And then you take a bucket of marbles and you dump it into the top of the board. So the marbles go bang, 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 and they all land on the bottom. What's the shape of the resulting marbles? What you always get is this lovely bell curve. It has a very particular shape to it. And so you can ask yourself, where is the shape of that bell curve encoded? And so you can, certainly it's not going to appear if you don't have the wood and the nails and the marbles, but you can study the material of the wood and the nails and everything else all day long. What you're not going to find is a direct encoding of that bell curve. So the real question in all of this is to go beyond the terminology. I mean, I'm much less interested in terminology and more interested in what kind of conceptual framework lets us do better experiments in order to get new capabilities. So if you want to make changes, if you want to induce repair, to fix birth defects, to normalize cancer, which are all things we've done that I didn't get a chance to talk about, the question is, are you better off thinking about the material, the hardware, or are you better off understanding the software that happens there and targeting that? And that's exactly what I was trying to argue, that all of modern molecular medicine has done some amazing things with respect to hardware, but most of what we are not able to do currently is precisely because we've stayed at that level, which computer science and information technology left behind in the 40s. So yes, the hardware is important, but we don't all program with a soldering iron. And that's exactly what I'm saying as far as these. The reason we were able to do some of the things that we've done, which had never been done before, is precisely because we were able to adopt conceptual frameworks from neuroscience and from other fields that really take very seriously the guidance of the final outcome, not by the hardware itself, but by the software components that are not actually directly encoded by the hardware, but of course require it to function in the physical world.
Thank you.
Thank you for a very interesting talk. My question was in relation to, I guess you said it's not genetic, it's more about the stimuli that you use to change how the cells function and turn into what they're supposed to be. I'm wondering, like with those frogs where you were able to reproduce most of a leg, if you mate generation 1 of the frogs, which were able to, using the stimuli, create this new leg, would generation 2 of the offspring, if you cut the leg, grow a new leg because you've reprogrammed those cells, and at this point it is the genetic, I know we're trying to understand the difference between genetics and these stimuli, but would generation 2 have the capability and the cell memory to regrow that leg?
Yeah, great question. We haven't done the experiment. I don't really predict that it would, because I mean, I could sort of make up a mechanism by which that would happen, but I doubt it would be actually that this would be transmitted to the next generation through the egg and sperm and so on. But there is an interesting example of what you're talking about, which is in these planaria. So we make these two-headed animals, again, perfectly normal genetics. See, that's another reason why the genome is insufficient for all this, because if you just sequence the genome of the two-headed and the one-headed form, they're identical. You would have no idea that you're looking at a two-headed worm. It's simply not in the genetics for that reason. Those two-headed worms, when they reproduce, so the way these worms reproduce is they tear themselves in half, right? So they just tear themselves in half and they regenerate. Now you got two worms. So that's their mode of reproduction. And under that mode of reproduction, the offspring do in fact inherit the two-headedness. So two-headed worms fission and they create more two-headed worms permanently. So you've made a permanent line of animals that have a different anatomical structure. And you can imagine, we could throw it in the Charles River. We wouldn't, but you could imagine throwing it in the Charles River here in Boston. And 100 years from now, some scientists come along and they scoop up some samples and they go, oh, cool, two-headed form and a one-headed form must be a speciation event. Let's sequence the genome and see where the speciation event happened. And there's no difference between it. That is not where it is. And so that kind of a thing does propagate because the animals are not squeezing through a single kind of a bottleneck of a single egg cell. So there's a chunk of the planarian which now remembers that it's two-headed, and that chunk regrows into a new animal. Can that pass through a single egg cell? I don't know. We have no evidence that it can. I could sort of imagine how it would happen, but we haven't shown anything like that yet.
Hi, thank you for your talk. My question is how you propose that this electrical information is integrated in these organisms. For context, I work on the PAC6 regulatory genome, so seeing these eyes kind of generate just solely from electrical stimulus kind of makes me think it's not the genes themselves, but the DNA outside these genes. But I would love to hear your thoughts on that.
Yeah, great, great question. So a couple of things. So first, just kind of a little inside baseball about PAC6. So in frog, and pardon me if you know all this, but in frog, PAC6 will only induce eyes in the anterior ectoderm, right? Just in the front of the head. And if you actually look at the developmental biology textbook, what they tell you is that, well, the anterior neuroectoderm are the only cells competent to become eyes because they probed it with the master eye gene PAC6. If you use PAC6 as your stimulus, it's true. Only the anterior neuroectoderm can form eyes. But if you probe the cells with a much more meaningful stimulus to them, which is these bioelectrical states, actually you find out that pretty much, I mean, we haven't found a region in the body that can't do it yet. So these competencies, what we think of as competencies, are really relative to how much we've understood the material we're working with, and the limitations of the material are often limitations of our understanding, not the actual competency of the material. So now how does it work? So what we do know for all of these cases is the molecular steps on a single cell level. So we know that when we change the voltage, we know the transduction machinery, we know how that voltage is converted to second messenger pathways, how those turn various genes on and off. Some of them have to do with other kinds of chromatin modifications. So we have all those steps. All of that stuff, while it's important to know, is really extremely unsatisfying, because the phenomenon itself is not a single cell phenomenon. So what I'm truly interested in is not tracking down how one cell's voltage turns on specific genes. That's just part of the, I mean, so it's an important part, but it's just part of the story, because what you really want to understand is, okay, but why is the whole thing scaled correctly? What's the difference between the hand and the foot? Why the shape of a particular structure? And so what we're doing now is we're using computational models of network computation, so not single cells, but networks of cells, that try to understand how the collective makes decisions about its size and shape and things like that. So so there are for sure molecular stories to be told about how the voltage turns genes on and off, but what you really want is an understanding of why a particular voltage kicks off eyes versus legs versus, and that whole conversation, like what happens when we induce these eye spots. There's a whole conversation that happens between those cells and their neighbors, because those cells are saying to their neighbors, help us make an eye. And so we also need to understand it's not enough to know why those cells turn on various genes. We need to know what are they saying to their neighbors, and how many, and how do they know how many of them there are. But those cells are actually saying back, no, you should not be an eye. You need to be normal skin. And that's a cancer suppression mechanism. The cells that have weird voltages get told by their neighbors through the gap junctions. They get communicated to try to normalize. So there's a whole battle that's going on of identities, and it's not an identity on a cell level. It's not stem cells trying to decide what cell type they should be. It's large regions saying we are going to be an eye versus we are going to be a leg or skin or whatever. So it's a very multi-scale problem, the same way that neuroscience is multi-scale, where, yeah, there are people who study synaptic proteins, but there are also people who study, psychiatric diseases and psychoanalysis and so on, and every step in between, all the network science and in between those two levels. This is going to be that kind of science, I think.
Thank you for your talk. I have two questions. One is, what is the most frustrating thing for you for this field to move forward, if it's not a secret? And the other thing, maybe I missed it, but did you mention how these electrical pathways are programmed in the beginning, like initially? And because you changed them, so there was something that gave them at birth?
Yeah, good question. So in the frog embryo, for example, we can watch them form from the moment of fertilization. So what happens is that the process of fertilization breaks symmetry, and we've characterized this from the very earliest stages, how the various ion channels that are present set up different kinds of electrical states that start to perform different computations and end up breaking symmetry, amplifying that symmetry, and then making decisions who's going to be left, who's going to be right, anterior, posterior, and so on. So it's a combination of ways. So where do these patterns come from? Well, it's a combination of emergence, in the same way that Turing patterns, right? So people who study chemical signals have these this formalism of Turing patterns, which break symmetry and create order out of well-mixed chemical media. So some of it is emergent. Some of it is, of course, constrained by the hardware. So you need to have the appropriate, as somebody was saying there, you need to have the right, the hardware that's sufficiently powerful to do it, which means voltage-gated ion channels and voltage-gated gap junctions and things like that. And part of it comes from wherever it is that mathematics comes from. I mean, this is really a philosophical question, but think about it. If evolution, once evolution discovers a voltage-gated ion channel, what you have there is a voltage-gated ion conductance, which is basically a transistor. If you have a couple of transistors, you can make a logic gate. Once you make a logic gate, you end up with a truth table that tells you how information is processed by that logic gate. You didn't have to evolve that truth table. The truth table is given to you for free, including things like the fact that the NAND gate is universal and various other cool computational properties. All of that you get for free. And you don't have to evolve it. You get it from wherever it is that the laws of computation, the laws of mathematics, the same way that if evolution discovers 2 angles of the correct triangle, it doesn't need to go look for the third. It can save all that time. You know what the third is going to be. So there are all of these interesting free lunches, in a sense, that evolution gets to exploit. And I don't know where they come from. Plato and Pythagoras thought that they come from this Platonic space of universal facts that exist separately from the physical world. That's as good an answer as any at this point.
Okay, we have many questions, so let's just keep the answers a little so we can get everyone here.
Hi, my name is Angeline. I'm currently studying neuroscience at Columbia, and I want to know, I'd like to know what you think about evolution as a mechanism of design. Like, for example, evolving a bot, like a biobot, instead of like designing it, or like both designing and evolving it. And like, would you edit Hox genes? Would you take advantage of epigenetics? And also, you sort of preempted this with your last answer. What do you think? How do you think we can take advantage of the vast computing potential embodied in biological components like membranes, like as you just remarked, like logic gates and ion voltage controlled ion channels are basically the same thing, and like there is a potential to program that.
Yeah. Okay. So there was a lot there, and I guess I was asked to keep my answers short, so I'll do my best and just pick one of the things you said. So we are doing a lot of work in programming both our Xenobots, which are the frog version, and the Anthrobots, which are the human version. We are not touching the Hox genes. We are not touching any of that stuff. We are learning to give them stimuli and take advantage of the cell's natural problem-solving capacities. We are training cells with rewards and punishments. We are trying to understand how they interpret various signals that cause them to acquire different shapes and functions, and we're trying to communicate that to the cells and let them figure out all the downstream molecular steps that need to happen to make it come true. I mean, the thing with evolution, I'll just say one quick thing about evolution, which is that I think evolution works completely differently with an agential medium like this that is able to solve problems than it does with the standard story of evolution, like over a passive medium where the genotype determines the phenotype. That middle software layer, that morphogenetic plasticity that allows it to solve problems, makes evolutionary dynamics completely different, because selection has a hard time seeing actual genomes. Then what it sees is the outcome of that competency process. And so you end up selecting for competency, not for actually better genomes. And that's an intelligence ratchet that goes all the way from developmental mechanisms all the way up, I think, to behavioral intelligence.
Thank you for the talk. I was wondering if you consider like moving the eye of the tadpole from where it normally is to the leg as a deviation from the norm. Is there a number of deviations that can be handled before the like mechanisms just break down? And what's that threshold look like if there is one?
We've never reached a threshold. I think that I don't think there's any kind of a threshold in that sense. I mean, you can certainly overwhelm metabolically. You can overwhelm the system. It needs a certain amount of energy just to undergo morphogenesis. And of course, that plasticity is not unlimited. I mean, there are many things you can do to screw it up so that it cannot fix itself. So birth defects are a thing, and that's because these are not infallible pathways. They adapt to a huge number of interventions, but of course, it's not unlimited. So I think the limitation is it's not the number of things you change, it's the kind of things, and whether or not you've broken the system's ability to remember what the right shape is and then act on it.
Hi, Michael. Fascinating topic. Thank you so much. So, what I would like to better understand is how do you think that such complex information as the performance of a task can be encoded outside of the nervous system? Since in the nervous system we have synapses, and the fundamentals of learning and memory, it's synaptic plasticity, potentiation and depression of synapses. So how do you think that such complex information can be encoded and modulated through simply ion channels that don't have synapses between them to communicate information?
Well, okay, a few things. First of all, I'm not suggesting that behavioral information it relies entirely on ion channels. That's not what I've said. But they do actually non-neural cells do have electrical synapses between them, gap junctions, which do store state and things like that. But overall, the story, and I know this is this kind of the standard story of neuroscience, that it's this, synaptic plasticity and LTP and so on. That story, not from my work, but from lots of other people's work, that story is significantly cracking at this point. And I would recommend the work of Sam Gershman, Randy Gallistil, David Glantzman to look at some alternatives, because I don't think that's, many people no longer think that that's the entire story. And overall, if I had to guess, I think that the vast majority of that machinery that you're talking about is not actually for storing memories. I think it's for interpreting them. And I think that there are numerous substrates within cells, neural and non-neural, that basically act like almost like a reservoir computing scenario. But a lot of the actual degrees of freedom are in chemical systems. They're not necessarily synaptic, and the neural networks are actually there to try to interpret it on the fly. So it's a long conversation to have, but look up some of those papers and you'll see this is not my idea. This has been around for some decades now.
Just keep your questions and answers. Like, we have like 10 more questions, so.
Hey, thank you so much for the talk. I'm curious on your thoughts about whether this electrical information can be accessible in higher order cognition. In humans, that might be in conscious perception, and other organisms that might look a little different.
Yeah, if I understood correctly, that's the project of neural decoding. So lots of neuroscientists work on this idea of reading out electrical information out of mammalian brains, or including human brains, to try and understand what the person is thinking.
Hi, thanks for the talk. I'm Kazra. I'm wondering, when you say all intelligence is collective intelligence, is there sort of a principled argument for that? Like, can we imagine intelligences that we haven't discovered that could not be collective? Like, why are all intelligences collective?
Well, as far as we know, so certainly on Earth, every intelligence is made of parts. I cannot, maybe somebody has better imagination than me, but I cannot imagine a truly sort of homogeneous monadic single object that is not made of parts that can change and that function relative to each other and so on, that could then learn and do the kinds of things we expect of intelligence. If it doesn't have internal parts that can change state, how is it learning anything, right? It's not. I just can't imagine how you could possibly have an intelligence that is one indivisible thing that does not have internal states that can change. Every intelligence that we've ever seen, every piece of active matter and intelligent living thing that we've ever seen is composed of parts. I just don't see how it would be otherwise.
Go.
Sorry. Hi. Great talk. So I was curious about, have you done RNA-seq before and after? For example, in that frog example, I'm curious if there are any transcriptomic changes that you were able to see, and if so, what are your thoughts on oncogenesis or oncogene activation?
Yeah, we absolutely, we've done tons of RNA-seq. There are massive transcriptional remodeling events that happen after the kinds of things that we do. Also naturally, so xenobots have a very different transcriptome from frog embryos. Anthrobots have an extremely different transcriptome from normal human, from the tissue that they came from, even though there are no genetic changes, no weird nanomaterials, no drugs, no transgenes, no synthetic circuits. So yeah, massive changes. And as far as oncogenes, the story that I didn't tell you is that there's a whole way to look at cancer as a kind of dissociative identity disorder of this collective intelligence. And what that means is that you can actually address it not by trying to kill the cells, but by trying to reconnect them back into the electrical networks. And so what we do is we inject human oncogenes like KRAS mutations and so on into these embryos. Normally they make tumors, but if we then manage the electrical connectivity correctly, even though the oncoprotein is blazingly expressed, you don't get tumors. You get perfectly healthy tissue with cells that are participating in the network activity and doing what they're supposed to do. So there is a cancer therapeutic story here as well.
Hello, Michael. Great talk. Thank you very much. So I'm a neurosurgeon. I have an interesting observation that you might find cool too. So we have had patients that we operate on, say, lose some function, speech, and then rehabilitate. After 6 months, they're back. They regain their speech back. And when we operate on the same patient again, the same spot where we lost speech last time is completely resectable and no function change occurs. So this is very rare. I saw this kind of phenomena happen 3-4 times, but I was and I'm not familiar with your field. But is there a paper you can direct us, something that that describes what you are describing here, the morpho memory and the regular memory? How do they work in the human brains?
Okay, there are lots of papers. How they work in human brains is very unclear because obviously experiments are really hard for ethical reasons. You can't do this in regular humans. There is some clinical data. Drop me an email, michael.levin@tufts.edu. Send me an email and I will shoot you some reprints of what we have.
Hi, Professor. What do you see is the biggest problem we need to solve before using this to program organisms?
The biggest problem is conceptual. The biggest problem is developing an understanding of how we are going to recognize and communicate with a collective intelligence, in the sense that we've done experiments with even gene regulatory networks without the rest of the cell, without changing promoters, without any of that, just by their dynamical properties, have 6 different kinds of memory that they can do, including Pavlovian conditioning, just GRNs on their own. And so that the ability to recognize that kind of property and to learn to engineer with an agential material, not the way we've engineered with passive materials, which we've done for millennia, but a material with an agenda, a material with a multi-scale set of goals and competencies and problem-solving skills. It's a completely different way of engineering. It's a communication and a collaboration with your material. It's not the kind of micromanaging that we've been doing for since the beginning of the scientific enterprise. So those issues are the biggest barrier.
Thanks. I noticed you guys mostly use like endothelial or epithelial tissues for like the xenobots, anthrobots, and from different species as well. Is that just sort of purely methodological opportunism, or is there like a relationship between behavioral plasticity and the characteristic time scale at which they divide and die and so on?
Yeah, interesting. No, I mean we've done everything. We've done mesoderm, endoderm. We've done human mesenchymal stem cells. The reason the bots are epithelial is because those are the tissues that are ciliated, and they have the little motile cilia, and that's that behavior that allows them to swim and to have visible behavior. Now, I also think that there are many interesting behaviors that happen in physiological and transcriptional spaces, but for our first kind of outing of this stuff, it was important to have behaviors that people can see and really recognize as behaviors. So for that, you need them to be motile, which means cilia, which means the epithelium.
Cool, thanks.
Hi, thanks for the talk. I really enjoyed it. I had a sort of question about the role of, you mentioned membranes were used to sort of predict the next downstream series of events and intracellular events that contribute to the gross development. What do you think of epigenetics and how you would sort of encode that in the computational model, and sort of what those next steps are, like methylation, alkylation, all those different types of things? Because you accounted for membranes, but not, I guess, those types of changes. And I guess more realistically, how do you account to encode those as next steps that a computational model might predict, or even downstream?
Yeah, we have them in our model. So we know there are acetylation and methylation events that are downstream of the things we do. It's there. By itself, it doesn't really add much. All of the kind of high level decisions about what's going to happen are made, at least as far as we've been able to see now, they're made at that electrophysiological level. All the gene expression changes, direct or indirect, through chromatin modification or downstream. Yeah, they're there. You can model them, but they're not a great target for modification. You know, if you want, I mean, there's certain things that you can do that way, but typically, if you want to make these large scale changes, you're much better off doing it as high on that control hierarchy as you can. By the time you get down to the chromatin modification, you are having to micromanage a lot of things to get what you want.
Hi, thank you for your talk. I was really interested in the conditions of this kind of initial bioelectrical field. You showed several examples of, for example, cells creating sending bioelectrical signals amongst each other in order to stimulate growth in a certain way. I'm wondering, when it came to maybe like the first organisms or organisms in a vacuum, are we kind of sending out certain frequencies or electrical stimuli in order to simulate that? Or what were those types of kind of initial field conditions?
Yeah, good question. I mean, we are living cells put out all kinds of things across the electromagnetic spectrum, ultra weak photon emission, all kinds of AC kinds of weak magnetic kinds of things. I don't study any of that, so I have no idea really what the implications of that would be. Everything that I've shown you was purely electrical. There's no magnetic component really here. No waves. This is purely electrical, and it propagates through water. So the stuff that I showed you doesn't really work through air. But having said that, it is true that living things do emit electromagnetic radiation, but I don't think we know what it does.
Thanks.
All right, last question.
Yeah, so thank you so much for this talk. Just a quick question about, like, because I guess you can kind of call it like a programming type of question. How would you like describe the type of like bugs that you encounter, so to speak, in this situation? So let's say you want to create like a worm with two heads, and like the outcome is just different than what you expect. Like, how do you? I guess first of all, I'm guessing you've seen that, and second, how do you like deal with quote unquote debugging that?
Yeah, great. The biggest issue here is that you have to think completely differently than you do with regular programming, because in regular programming, at least most of the time, you're dealing with an obedient passive medium. So when you have a command, whatever that command may be, you are pretty much guaranteed that the hardware is going to carry it out. It may be the right command, it may be the wrong command, but when you say, add these two numbers, if your hardware is not actually going to do it, then you really don't have a good computer, right? The whole point of hardware is to isolate those layers so that at the higher level you don't need to worry about where the electrons are going. You can assume that the chip is doing what it needs to do and it's going to get carried out. Biology isn't like that at all. So what's happening here is that every level has its own agenda. So when you are providing certain signals, what you end up with are targets that either aren't paying attention, or they are paying attention and they refuse, or they actually fight you on it because they're going to do something different, or they have some other thing that they want to do. And this happens at every level. It's not just behavioral scientists know about this because it's really hard to do experiments because of individuality. Cells do this too. So you're dealing with a material that is not a passive material that has well-separated levels of abstraction where each level can depend on the bottom level to be perfectly obedient. They are all pursuing their own agendas. They are cooperating, competing, communicating with each other, interpreting each other. The computational paradigm that Josh Bongard and I have come up with for this is called polycomputing, and it's simply the idea that you have a huge number of coexisting interpenetrating agents which are all interpreting and hacking each other in different ways. So there's not a single interpretation of what an event is actually computing. In our computing architectures, we all agree when there's an AND gate, we all agree this set of physical events, that's an AND gate, it's computing it. In biology, you have lots of little observers inside the cells and the tissues that are all interpreting these events as completely different computations. And we as a third person, sort of third party observer, often have absolutely no clue what they're doing. And that's really hard. It's a very different way of programming.
Amazing. Well, what an amazing talk. And thank you so much let's give
Thank you, everybody.
Professor Levin another round of applause.
Yeah, thank you so much. Great questions. I really enjoyed the discussion. Yeah, awesome.
Okay, now we will also talk a little bit about decentralized science. And I'll follow up with, you're welcome to stay or whatever's good. So let me share this tab here.
Thanks, everybody. I've got to run. It's been a long day. I got to go.
Thank you.
Thanks a lot. Yeah, great discussion. Thank you. Bye.
Okay, so let me switch over here. So, okay, oh yeah, so first off, decentralizing science is important. So the first part of decentralizing science is attracting more people. So if you enjoyed this event or you wanted to share it with another person, make sure to post it. It really helps us. You can take out your phone. I hate to be annoying about it, but everyone should take out their phone and take a picture and send it out to other people. Even if it seems weird now, but just it helps. We have information about. I'm going to give you information on our next. Oh, first we want to thank the people who gave us this space today, which is Alchemy. And they are the most reliable way to build Web3 apps. Specifically, Nikhil, a college friend, was very kind to give us the space, donate the space. So this is the DeSciNYC community. We have lots and lots of speakers. This is the 14th one we've done. We've had people on mushrooms and blinded studies and bees and AI and NFTs, all sorts of things. And we have another one. Oh, we have a community if you want to join. See, now people take out their phone. We have a Telegram group so you can stay in touch after the event with each other. Just giving another hint here if you have your phone out now. Okay. We have a website, so DeSci.NYC. We have all our previous events there. We have the videos. We record everything. We post it online. We have swag you can get, other things like that.
Can you go back to the Telegram?
Oh, yeah. Cool. Okay. This is our next event on August 1st. It's going to be amazing. There's going to be, it's on this guy who's doing this it's Super Size Me. Remember that movie with McDonald's? And he's going to be doing Super Size Me, but with wearables. It's going to be really cool. I spoke with him a few times, and he's an interesting fellow. and he's developing like a new type of wearable for your toilet, and and he's trying to like destroy his gut right now and then heal it and then track the process, and we're gonna be the first ones to hear about his poo. Oh, this is one thing we're trying to do more of, like having like a volunteer group. Right now, it's sort of like a skeleton crew that's doing this. Oh, thank you, Brian, for helping tonight. Thank you, round of applause, Brian. And Lev as well on the video. And Brian checking. And Brian on the code also. And so if you're interested in volunteering for the next event here, we'll get dinner, we'll assign roles, and you can help out, be more part of the crew and pick speakers. What was it?
A little after April.
I forgot to update this slide. Okay, so now I'm going to talk about like what is decentralized? I wanted to do this talk before, but he had a tight constraint, Professor Levin. So we'll do this talk after. And I want to just say, like, why is it important as a society and as like humanity that we like decentralize science? So, like, imagine like like right now, universities are like science is like hugely beneficial to humankind, right? So like that's probably one of the best metrics of progress. And right now, like a lot of it's done in like research and development labs, and it's done in like universities. But the quality of the research in these things is very tightly kept. It's a secret, and also that sometimes the quality of like academic research is really bad, and it's like not reproducible. And imagine a world in which like the internet was like a giant lab where people could like contribute data to it and get rewarded for it. And I think that like this is not too far off. I think like universities are not as important as they were 30 years ago. And where is that science and innovation going to happen? It's going to happen on the internet. And this is like extremely valuable, and the people that participate in it should get rewarded for it. So with this sort of premise is like, if you had this as your goal, what would be the easiest way to do it is you would use this blockchain instead of just for like financial incentives, for like swapping tokens, and like you swap some Bitcoin for some Ethereum on Uniswap, which is cool, but like the underlying value attached to each of the tokens is like sort of arbitrary. Whereas if you like instead use the blockchain to assign, each token were assigned to a research project or to intellectual property or to a new drug or to bioelectricity, and then you allowed people to like swap those types of tokens for the IP that's attached to those or contribute to a research project. This could be really cool and make new types of projects happen and maybe fund things. And I want to emphasize that like this isn't far off. I don't think that we're that far off from this sort of thing existing in the world. I think it could be within the next like 2 to 5 years. I think it's very possible. And then the other big thing that's changing around like why this is possible now is like AI is like going to be a scientist very shortly. So the idea that you're a PhD student and then you have 6 years of knowledge and 4 years of undergraduate in this very specific domain is like passé. And now anyone can be a research scientist with just like access to ChatGPT, and you can get up to speed very quickly. So in this like new decentralized science thing, I think it's possible to have like 1,000 times the number of scientists that exist today, because there's a lot of people who aren't part of a university who want to become like a scientist. People will be incentivized so that you don't have to do it through like you get a crappy salary at a university. Like no one wants that. Like people want to be like incentivized to do really great work. And so I think that this is very possible to have like wealthy scientists and a lot more of them, and like similar to how like maybe like 30 years ago or 40 years ago, like there was only a couple computer programmers, but then like computers became cheaper, GitHub became available, tools became easier to use, and then there's like everyone is a programmer today. And I think the same thing is we're on the cusp of that for science. So it's like what DeSci is going to do for science. Like it's going to decentralize science. But how many people know what DeFi is, just in general? Decentralized. Okay, a lot. DeFi is like decentralizing finance. So that's like the idea that instead of like opening a bank account at Wells Fargo, there's probably a lot of people who can't open a bank account who would like to be banked. And so having like an Ethereum a Bitcoin wallet address or an Ethereum address and allowing them to swap different tokens opened this up to a lot more people and also removed a lot of the middlemen. So here's a little table I made. We call this traditional science on the right and decentralized science on the left. And I start off with, this is just sort of the process of a typical science application. So the first thing is funding. So I hate funding because like normally if you're not getting funded as a scientist, it means like, I've never seen a really talented scientist complain about funding. So it means like something else is wrong, but it's the first thing here, so we'll start with it. So the funding is like, a lot of funding happens because governments fund a lot of research. So like the National Science Foundation or the National Institute of Health. And the problem with that is like, they just fund very specific things and they don't go broad and they only fund things that they know. They don't fund moonshot ideas. But in DeSci, you have access to a lot more people. So you can crowdfund ideas and you can do a lot more crazy stuff. Publications are very broken in traditional science. So publications in traditional science that go through a peer review, but the peer reviewers don't actually read the papers. They just sort of look at it and move on. In DeSci, anyone can be a peer reviewer and you can like bet on individual contributions. Then data sharing. So in traditional science, data is very locked. And in DeSci, the data sets are like open and you can pay and reproduce experiments very easily. Community, like in a traditional science setting, like you only sort of do research within the lab setting. But in DeSci, you have a global access. You can access a lot more people, which is cool. IP can be shared. Governance is, instead of just like the lab manager doing all the research and directing things, everything can go up to a token vote. So if there's a question around should we do experiment A or experiment B, instead of just relying on one person to do it, you can have like a vote on a snapshot and decide on what, then use the collective intelligence to figure out what the next step should be. And then the reproducibility, I think, is one of the most important ones. Like, I think in a lot of research today, like most of the papers are not reproducible because they're either too complex, they don't reveal enough about how their methodology, or they p-value hack, and there's all sorts of issues. But in DeSci, because everything is disintermediated between physical entities, everything is sort of logged on chain. So you can easily just like replay back everything and see which science is valid and which science is invalid. Okay. So the way it works is like, if you want to create a DeSci project, you have an idea and you say, I want to create an idea around sleep. And everyone who contributes to that project is issued a token. So maybe you contribute through giving sleep data. Maybe you contribute through doing developing some sort of algorithm. Maybe you contribute through recruiting people. And for each of these different things, you get tokens. And we have the data pools, so people can sort of contribute to a data pool. So if, for example, for sleep, you can use like the Aura ring and contribute to a data pool, and that data pool will then be accessible to anyone who's interested in sleep. Other types of data pools that I think are really interesting are like Blueprint is very popular these days. So like longevity, like how do you determine if these types of things, these interventions work? Is you're going to need like very, very like broad data sets. So things around like food, things around Aura ring, and things around like genomic data are all going to be really, really interesting to determine like these are prime data sets to figure out like what's working and what's not working. You can do a lot more with like data quality. So right now in a research project, people just sort of like put in things, and like maybe the PhD student will like look for outliers and remove those and then sort of move on. But when you have like a decentralized science system, people can like stake on data. So when you submit data to a research project, you can submit like $5 along with it. And why that's important is because if you contributed bad data, like you can get slashed. So you won't get your $5 back. So you sort of like spam the system, and then your data will be removed. And so it's incentivizing you to produce like high quality data that can be verified. And that's like these are the types of things that you can do with like DeSci, but not with like traditional science. People can just like, you could have like an exchange. So like Coinbase, you could have like, oh, I want to invest in, or I want to invest, but I want to participate in like Bitcoin or crypto or like Ethereum. And similarly, it could be like, oh, I want to participate in an HRV study or a sleep study or a CO2 study. So it sort of opens it up to a lot more people. And then this is one of the more interesting things. You can like, instead of like, how many people know what a prediction market is? Okay, so imagine a prediction market. It's just like a betting market, or like you predict what's going to happen in the future. And if you're right, you win tokens, and if you're wrong, you lose tokens. And imagine like a prediction market for like scientific experiments. So you're incentivized to think about like which types of experiments are going to work and which type of experiments are not going to work. And this is very different from peer review. So peer review is like, I have no skin in the game. I don't care if this paper is accepted or rejected. Now let's like flip that around. If I'm correct, this paper is correct, I'm gonna like earn tokens. Now I'm like gonna spend a little bit more time reviewing it, because it's like you want to do really good diligence on it. So the upshot of this is like you're trying to align incentives between like high quality scientific research and like the outcome for the individual person. Okay, so basically, like, we're trying to build a platform that will like enable people to participate in decentralized project or to spin up one. We've started it already. It's called waterbear.science, and we're hiring. So if people here are interested in decentralizing science and like participate with specific roles we need are like we need like a BD and a community manager person, since it's a little we have like a lot of people inbound, and we can't really respond. So we're trying to grow. We're VC backed by like some of the best investors in crypto. So if you're interested, we're hiring. Here's our application. It's very short. We're in person. We work like normal hours, but it's all in person, which I think is good. And I think this is like a great community to pull from. So if you enjoyed this event and you want to participate more, definitely apply. And then, oh, that's it. Do you have any questions? Yeah. Oh, DeSci.NYC. Yeah. Yeah.
So I have a question. So normally the government, some of the biggest sources of funding for science and government comes from DoD, defense, energy, and medical. And a lot of times they put that science right into applications, which generates return for them. What do you see as the biggest drivers for DeSci in terms of funding?
Yeah, I think there's a lot of people that are just passionate about things. Like, I would fund Michael Levin researchers because I think it's cool. Like, I would much rather give him money than like some other like random like charity. So I think that's one. I think people that have a specific need for it, some like a rare disease, like people are much more interested in participating in that sort of thing. And if you give them like a pathway to back credible people and participate in that type of research, I think that's another one. And then people who are just interested in science as well. Like, I think there's a lot of people that want to continue education, maybe have knowledge, and they enjoy doing it in their free time. And also, I think the costs are decreasing. So like, previously it used to be a lot harder to do a science project, but there's a lot of like clinical research organizations and a lot of automation and a lot of AI that's happening that's, I think, bringing down some of the costs associated with like traditional research.
I mean, I guess following up on that. Yeah, good. I guess it's kind of like a follow-up, but there's usually a lot of IP coming out of research too, and spin-offs and startups and so on. I assume that could be also a framework where incentives could be aligned. If you get part of that, is that part of the entire equation, or?
Yeah, so that's like how it works. Is you have an idea around, like, let's just take an example. of like mental health. Like, so you create a mental health DAO around a specific topic. And then everyone who participates, like that sort of wraps, that's the IP. Any IP that's created as part of that group is tokenized, sort of. And everyone who participates and does work earns tokens based on their participation. So if you contribute data, if you contribute analysis, if you recruit patients, if you publish it, you become, you get these tokens commensurate with your, how much you contributed. Yeah. And so that's one of the things that's nice about DeSci is like, it solves the IP issue. So in like a traditional setting, you'd work with the university's tech transfer office, and they take a third and a third and a third, and it becomes impossible. And a lot of research dies there for partially that reason. But here it's much easier. Like, all the data is tokenized, and there's a lot of liquidity around it, so you can exit or enter back in if you choose. You can also like what's the word I'm looking for? Vest some of the IP, so it keeps people invested in the project. Like one of the problems now with like university research is people graduate and then they leave, and like, but if they vest it over maybe a longer vesting period, they might be more incentivized to stick around.
Thanks, Michael. You mentioned that if someone spams the system with incorrect data, that could be removed. How would that happen if it's on blockchain?
Yeah, I mean, it would just be marked. There's a couple different ways of doing it. One is to allow any data and allow like some agent to just, it depends on the research project. The simplest thing is allow all data and then use some sort of AI or human to judge which is good and bad quality data. The second one is to do like a community voting. So you have like a jury and they vote on each piece of data to see if it should be accepted or rejected. Or maybe you could use something like a liquid democracy approach where you can assign your voting power to another person and they can vote on your behalf of if this data is good or not, and you just trust this person. And then the crypto economic way of doing it is to have people stake money on their data. So before you can contribute the data to the data set, you contribute it along with a $5 bill. If your data is accepted by the community, you get your $5 back, you get your data accepted into the community and your tokens in return, and whatever benefits are associated with those tokens. But if your data is considered bad by some standard, then the $5 is removed, is kept by the community, and the data is thrown away. And the next question you'd ask is like, what prevents, like, why would you contribute to, what if there's a bad community and they just kept all the money associated with the data that's come in? And the answer to that is like, all transactions that have been submitted to the DAO to accept the data are on chain, so you could review the previous submissions and say like, they've submitted, 100 people have submitted great quality data and it's all been rejected. This DAO is terrible, I'm not gonna participate in it. Yeah. Anybody else? Cool, well, thank you all for coming. It's amazing to see all you here. And I hope to see you on August 1st. We might do one in between. We haven't scheduled it yet, but the August 1st one is the next one. And if you're interested in decentralized science, or you know someone that is, share it with a friend and recommend them. All right, thank you guys.